In this video, we will discuss about Haemophilus influenzae. Morphology, they are small gram-negative bacilli. Haemophilus influenzae, small gram-negative bacilli, which are non-motile, non-sporing. They are pleomorphic. That means don't have any particular shape. In sputum, they are seen as cluster of coccolobacillary form. In sputum, they appear cluster of coccolobacillary form. In the sample of CSF, they are seen long bacillary and filamentous form. CSF, they are seen long bacillary filamentous form. So, this one is a pleomorphic, don't have any particular shape. In sputum sample, they appear cluster of coccolobacillary form. In CSF, long bacillary filamentous form. Now coming to antigenic properties. There are three main surface antigen. They are capsular polysaccharide, outer membrane and protein antigen OMP, and lipo-oligosaccharide LOS. Three main surface antigens: capsular polysaccharide, outer membrane and protein antigen, then lipo-oligosaccharide. These three are main surface antigen. In this, we will discuss about capsular polysaccharide in detail. The type B capsular polysaccharide has a unique chemical structure which is made up of pentose sugar, ribose and ribitol. The type B, Haemophilus influenzae type B, capsular polysaccharide, unique chemical structure. This one made up of ribose and ribitol. The capsular polyribosyl ribitol phosphate antigen, which is presented in Haemophilus influenzae type B, which induce IgG, IgM, IgA antibodies, which are bactericidal, opsonic and productive. So the Haemophilus influenzae type B capsular polyribosyl ribitol phosphate antigen, which is presented in Haemophilus influenzae type B, which induces IgG, IgM and IgA antibodies, which are bactericidal, opsonic and productive. Haemophilus influenza strains which are lacking a capsule, so it is called non-typable strain. It don't have any capsule, so the strains is called non-typable strain. Coming to pathogenicity. This Haemophilus influenzae mainly affected human pathogen. They produced two types of infection, invasive or non-invasive. Invasive pathogenicity here, the bacillus itself primary pathogen, which causes acute invasive infection. Second one is non-invasive. This bacillus spread by local invasion along the mucosal surface. This causes secondary or super added infection, usually respiratory tract infection. Invasive infection occur in children. It most commonly by capsulated strain type B Haemophilus influenzae. The most important infection caused Haemophilus meningitis. Non-invasive infection include otitis media, sinusitis. The other diseases caused by invasive laryngoepiglottis, conjunctivitis, bacteremia, pneumonia, arthritis and pericarditis. Coming to clinical presentation, meningitis the most serious disease in which mortality up to 90%. The bacillus enter nasopharynx. From this nasopharynx through the bloodstream they causes meninges, inflammation in meninges. The most commonly occur in children. This one mainly affected in the children the age group of 2 months to 3 years. Laryngoepiglottitis, which is acute inflammation of epiglottitis with obstructive laryngitis. So it got that name laryngoepiglottitis. This is seen in children more than 2 years of age group. If untreated, this is fatal within hour. So this bacillus enter nasopharynx. After that it will reach to bloodstream. Then it will affected brain part. It mainly affected in children 2 months to 3 years of age. Next, tracheostomy to relieve obstruction by enlarged uvula. Here the uvula enlarged, so obstruction of trachea, so tracheostomy performed. Next one is pneumonia occur in infant. It is accompanied by empyema and meningitis. In older children and adult, lobar pneumonia occur, which is primary infection due to capsulated strain like type B Haemophilus influenzae. Next, bronchopneumonia may occur as a secondary infection with non-capsulated strain. Next one is suppurative lesion. It is first forming lesion like arthritis, endocarditis and pericarditis. These result from heterogeneous dissemination. Next, otitis media can occur by direct spread from nasopharynx. And there is buccal periorbital cellulitis in young children. Next one is bronchitis. The Haemophilus influenzae is associated with pneumococci and they causes acute exacerbation of chronic bronchitis and bronchiolitis. Moving to laboratory diagnosis. Collection of specimen which included CSF, blood or sputum. Depending on site of infection, we collected that sample. Microscopy. In the case of meningitis, it is a pleomorphic gram-negative bacilli are visualized under microscope in CSF. Third one is antigen detection. CSF is detected in the case of meningitis and urine in the case of systemic infection. The direct antigen detection is performed by lactase agglutination test. Then culture performed by using chocolate agar medium, blood agar medium with Staphylococcus aureus, then Levinthal medium, then Feltus agar medium. These are the different culture medium we are using: chocolate agar, blood agar, Levinthal medium and Feltus medium. This is a plate of blood agar in which Staphylococcus aureus is streaked across the plate. Staphylococcus aureus streaked across the plate. The specimen containing Haemophilus influenza has been inoculated. After overnight inoculation, the colonies of Haemophilus influenza will be large and smaller farther away, decreases size farther away, and this phenomenon is called as satellism. The plate is the blood agar medium. In this central we streak the Staphylococcus aureus across the plate. Then Staphylococcus aureus streak across the plate on blood agar on which the specimen containing Haemophilus influenzae has been inoculated. After overnight inoculation, the colony of Haemophilus influenza will be large. Colony of Haemophilus influenza large, well developed along the side the stick of Staphylococcus. Here Staphylococcus, here larger colony, then smaller colony here, there away. This phenomenon name is called as satellism. Next, biochemical reaction. It ferment glucose xylose with acid production, but lactose, sucrose, mannitol not fermented. Ferment only glucose xylose, not ferment lactose, sucrose, mannitol. It shows catalase positive, oxidase positive. It can reduce nitrate to nitrate. So catalase positive, oxidase test positive. It can reduce nitrate to nitrate. Then treatment. For treatment purpose, we are using cefotaxime or ceftazidime. Amoxicillin also we are using. Then preventive method. Prevention is by active immunization with Haemophilus influenza type B vaccine or HIB, Haemophilus influenza type B PRP vaccine also we are using. So this is all about Haemophilus influenzae. Thanks for watching.